Okay, cool. My name's Matt Zuellig, and I work with Andrew Swagger back there, and I'm studying genetic incompatibility system in Mimulus. So the lab is really focused on understanding, like a lot of people in this room, how do new species evolve, and as we know, a lot of us study that in the context of how does reproductive isolation evolve in lineages, in populations over time. Allowing them to diverge and accumulate barriers to gene flow through this process of divergence. So there are really two varieties. The prezygotic barriers can largely be seen as adaptive changes within lineages over time, or at least changes that can respond to some kind of selection within lineages that prevent them from mating upon secondary contact. But alternatively, there are postzygotic barriers that cause things like hybrid problems, and it's not as clear to understand how a selection could act to cause things like hybrid inviability or sterility. So we're largely focused on studying the genetic basis and the evolutionary dynamics of postzygotic barriers in the lab. And postzygotic barriers are largely caused by Dobzhansky-Moeller incompatibility. So here is the basic two-locus model, where in lineage 1, these two genes interact epistatically with one another. So in lineage A, we get little a evolving on the big B background and little b over here. And over time, maybe those little a's and b's go to fixation, but when they come back together in this hybrid background, they haven't co-evolved, and therefore there's some bad thing that happens, maybe sterility. And so we're interested in what types of genes, are there certain classes of genes that cause hybrid incompatibilities? What are their molecular functions? Are there common architectures for certain ages of species? Things like that. In addition, we're interested at this level here. Not all incompatibilities are fixed. There are things happening in populations that cause alleles to increase and decrease in frequency. And so we're interested in what are the forces that allow for, say, little a to increase in frequency over time. Where did little a come from? Did it arise in this lineage, or did it arise in some ancestral population? And also on secondary contact, how do we maintain little a and little b in this scenario in a hybrid context? When species are hybridizing, why don't we just basically intergress, say, big A from over here and replace that bad little a that's causing hybrid problems? I'm studying incompatibilities in the Mimulus guttatus complex, and really just two species shown here, Mimulus guttatus and Mimulus nisutus. So you can see that guttatus is everywhere in the shaded region, and Mimulus nisutus is nested within that range. And these are cool species to study speciation with because they're very recently diverged, between 250 and 500,000 years ago. And there are a number of different reproductive barriers within the system. So there are prezygotic barriers, they prefer different habitats, they have different mating systems. Guttatus being a large outcrosser and this one being a sulfur. They flower at different times, but there are also a number of postzygotic barriers such as hybrid sterility and hybrid inviability. And a lot of those barriers are polymorphic within at least Mimulus guttatus. So there's a lot of variation to select on within these systems. Despite all these barriers, reproductive barriers, they're still in sympatric populations where you see guttatus and nisutus really close to each other. A low amount, but still an observable amount of hybridization and introgression. And introgression in this system largely goes from Mimulus nisutus to Mimulus guttatus. So I've been studying this character called hybrid lethality, and it's manifest as these white seedlings shown here. And what happens is basically these are hybrids between the two species, and these little guys don't make any chlorophyll, so they are going to die very soon after taking this photo. We produced them by crossing Mimulus guttatus and Mimulus nisutus and producing an F1, and then we self that and produce the F2. And as it turns out, and I've characterized a lot of this, I'm running over a lot of mapping stuff that I've done, but 1/16 of the F2 seedlings are white. And there are 2 nuclear recessive loci that cause this. So you can perform the cross reciprocally and see the same phenotype caused by the same loci. And I mapped both of those loci using bulk segregant experiments and F2 recombinants. And I have like exciting, cool new mapping results, but I can't discuss those today. So the punchline from this little part is to say that I have 2 loci that I call hybrid lethal 13, because it's on chromosome 13, and hybrid lethal 14. And those are the Gutatus and Nasutus components of this DMI in Mimulus. So for the visual people, you can see you have your F1 heterozygote, and you become 1/16 of F2s will be homozygous at 13 and 14 respectively, and that causes white guys. So we can genotype markers flanking both of those loci to characterize the alleles within populations throughout the range. And that's what I am going to talk about next. So we were interested in understanding what is the distribution of hybrid lethality alleles within both of these species. And so we're wondering, like, are hybrid lethality alleles fixed or polymorphic within species or even within populations? And are hybrid lethalities locally distributed, or are they generally just widespread throughout the range? Here is a map of allele frequencies for hybrid lethal 13, the Gutatus, what we found to be the Gutatus component. And over here are like the global frequencies of hybrid lethal 13 and Gutatus and Nasutus. So every circle represents population. Circles that are yellow are Gutatus, blue wrapped circles are Nasutus, white equals an incompatibility allele, and green shading equals a compatible version of that allele. And the big punchline here, as you can see, we sample not a ton of populations all over the place. It's very dense in California, but the hybrid lethal 13 alleles are widespread in the system, maybe suggesting that they're an older allele. And they are polymorphic within Gutatus, and they're very overrepresented in Gutatus in 70% of alleles sampled. And they're also in Nasutus, but at a very low frequency of 13%. So there is polymorphism for hybrid lethal 13 in both species. Next is the other allele that I originally found in Mimulus Nasutus, and here again you can see that the alleles are widespread throughout the range. And Mimulus Gutatus, though, you basically see the flip here. That Mimulus Gutatus has a frequency of the incompatible allele at about 28%, and Nasutus at about 69%, so relatively high. So we can see that there's a lot of polymorphism there, and that these alleles are very widespread in the system. Additionally, we looked at the populations of Mimulus that carry both copies of the allele. So we're interested in in what context can these alleles potentially interact? And in 7 out of the 18 populations of Mimulus Gutatus that we studied, we found that Gutatus carries both hybrid lethal 13 and hybrid lethal 14, setting the stage for incompatibilities to act potentially both interspecifically between Nasutus and Gutatus and as an intraspecific problem just within Gutatus. And to get an assessment of whether we actually ever see that, we took wild collected individuals and self-fertilized them, planted that self-seed, and just saw if there were any white progeny in the self-individuals to see if the hybrid incompatibility was like present in full in individuals. And in 15 of 69 Mimulus Gutatus individuals, we observed exactly that, attributable to this incompatibility. So that's a very large frequency of the full incompatibility existing within individuals in the wild. And I just recently planted out a bunch of wild collected seed, and again we're observing white seedlings that were actually produced in the wild, but we need to confirm whether that's due to this incompatibility or maybe something else that causes hybrid lethality problems. So the punchline here is the incompatibility is very active in the wild, it seems. It's very likely. So here, in the future, we want to ask some basic questions about what are the sources of polymorphism at hybrid incompatibility alleles. So here you can see that this would be hybrid lethal 14 diverging in Guttatus and Nasutus, and same with hybrid lethal 13 here. And we're interested in why do we have this polymorphism at loci. And one reasonable hypothesis is that there's simply some unidirectional introgression from Nasutus contributing to the Guttatus gene pool. So these are recessive alleles, and it'd be maybe relatively easy for alleles from here to introgress into the Guttatus background. And this would ultimately lead to the demise of the hybrid incompatibility in the system. So we would predict that putatively introgressive alleles will exhibit variation more similar to heterospecifics than conspecifics. But alternatively, this figure is might get good, but this is the case for incomplete lineage sorting, where perhaps there was an ancestral gene pool of hybrid lethal 13 and 14 carrying Guttatus-like ancestors, and the alleles simply haven't sorted out yet within either of these species, and so maybe gene flow is very limited between these Guttatus and Nasutus, and we just haven't resolved this yet. In this case, we predict that compatible and incompatible alleles will be more genetically similar within conspecifics than between heterospecifics. Is it over? Okay. Well, it's basic. It's real close. Okay. So the conclusions here are that hybrid lethality is genetically simple. It's a real tractable system to study. And the lethality alleles, I've been saying, are very widespread, and they're polymorphic within both species. Additionally, hybrid lethality is expressed in natural populations, and that makes this a really excellent system for studying the dynamics of hybrid incompatibilities in real-world systems where DMIs might actually be important in the speciation process. So with that, I'll thank all these folks and funding sources, and I will take any questions. Thank you.